Good day everyone, welcome to the course on next generation sequencing technologies, data analysis and applications. In the last few classes we have covered the preliminary steps for transcriptomic data analysis. So, namely we have talked about transcriptome assembly, we have talked about normalization, and now we are ready for the main analysis, right. This is a very popular analysis that we do on transcriptomic data sets and the analysis is called differential gene expression analysis. So, this is the agenda for today's class. We will talk about differential gene expression analysis, we will talk about the concepts, and then we will introduce some of the tools that we use for doing this kind of analysis. Okay. So, let us get started. Here are the keywords for this class that will come across these terms. First one is negative binomial distribution, dispersion and hypothesis testing. Okay, so just to remind you about the steps of RNA-seq data processing pipeline, we have covered through the mapping, read mapping, and for transcriptome we have talked about the challenges. Then we have talked about assembly that we can do and we sometimes do. Then of course, we have the quantification step and followed by normalization. So, in the last class we talked about different types of normalization which can address different technical issues that are present in the read data. Now, we are ready for the analysis part, right, and this is what we are going to discuss today. Okay, so, the analysis is differential gene expression analysis. This is a very popular analysis that we do, and the goal is to identify genes that show difference in expression between two sets of samples. Okay, so, these two sets of samples could be a comparison between control and treatment groups, right. So, imagine you are doing this experiment on a control group, you want to test, right, how a treatment affects the cell behavior, cell physiology, etc. So, you have control cells and then you have the cells that are treated with a drug or some other type of treatment, and then you look at the change in expression of genes, okay, and you want to identify which genes are responding, because that might give you some insights about the function of the treatment that you are using. We can also do this kind of analysis for disease versus healthy samples, right. So, you can imagine we have, let us say, disease tissue, and the most common example that we use is the tumor tissue, right. So, we have tumor samples and then we have the healthy samples, right. So, we can compare the transcriptome of tumor versus normal cells or normal samples, and we can identify genes that show difference in expression in tumor cells. Okay. So, this is something again will tell us about the biology of tumor, right. So, how the tumor progresses and how all the stages are developed, right. We can also do this for simply studying gene expression pattern across two different conditions. So, you can think about cells that are in normal healthy condition, right, and then you kind of expose them to some sort of stress. Okay. Maybe this could be something like an oxidative stress or maybe salt stress, those kind of environmental stresses that are commonly found, right. And then you can ask, right, which genes are overexpressed when we expose the cells to the stress, right. So, again, if by identifying genes that show difference in expression in the stress environment, you can identify genes that are actually involved in the process of stress response. Okay. So, all this analysis, right, and all these examples that I gave, the goal is to identify this class of genes, so that we learn more about the biology of the system, right. So, what is actually happening inside the cell, how genes are responding, which genes are responsible for what kind of task, etc., those kind of goal we have at the end. Okay. By the way, so this analysis can be done, of course, not just for comparison between two sets of samples, you can do this for multi-class multiple sets of data, right. So, in case of disease versus healthy, you can think of, for example, you have tumor, you have healthy, you have, let us say, other types of samples, right, other diseases, not just tumor, maybe some other disease, right. So, those kind of analysis can be done, and similarly for condition analysis, you can have multiple conditions, right. It is not just condition 1 versus condition 2, you can have, let us say, normal condition, then you have, let us say, salt stress, you have oxidative stress, maybe DNA damage, all those conditions, and you can do all these comparisons as well. Okay. All right. So, before we actually proceed to differential gene expression analysis, right. So, we sometimes prefer to perform something called preliminary analysis, right. So, these preliminary analysis help us identify, okay, which groups should we compare, okay, and which groups are similar in expression to each other, right. So, we have the normalized count data which we can use to actually do this preliminary analysis. Okay. So, I will talk about two different preliminary analysis and commonly used, but you can of course think of other ways of looking at the data, and there are of course many innovative ways people are coming up for this analysis. Okay. So, we can identify the most appropriate groups for comparison, right. So, let us say we identify where these groups are very different and would be most interesting to study, right, rather than groups that are actually very similar to each other. And so, one of the first method is the distance or correlation based clustering. So, we can cluster the samples based on their normalized count data and see if which samples cluster together and which samples are apart, far apart in terms of their expression, right. So, again if we want to do differential expression analysis, it would make sense to compare samples that are actually far apart, right, not the ones that are very similar to each other. And then we have another method which is called the principal component analysis or PCA. Again the idea is very similar, right. So, we want to actually look into the samples that are actually similar to each other and the samples that are different from each other. Okay. So, I will just give you some examples with some sample data and will show you how these will look like. Okay. So, I have been talking about these 16 samples, right. So, we use this data for normalization, we explored different types of normalization with that data. So, I will use the same data for actually showing you how this correlation based clustering or distance based clustering will look like, and if we do the principal component analysis on that data set, what we will get, right. So, that will again give us now some insight into the data, okay, and the biology of the data. Okay. So, let us look at the preliminary distance based clustering, right. So, here what happens is that we calculate something like a distance between the samples, right, based on their normalized count data, and we do something called hierarchical clustering, and what it achieves is that it will group similar samples in cluster, right. So, they will occur together in the cluster. Okay. So, again we will see this now with the example and you will probably better understand what we mean by this. Okay. Okay. So, here are the 16 samples and they are in 4 groups. Okay. So, we have group 1, group 2, group 3, group 4. You can imagine those as 4 different conditions, right, and so we want to see, right, which conditions are giving similar transcriptomic profile and which conditions are giving different transcriptomic profile, because based on this analysis we can decide, okay, maybe this is the most interesting comparison for differential expression analysis. Okay. So, what you see in this plot here, okay. So, here are the samples, right. So, along the rows you have the samples as well as you have along the columns you have the samples, right. So, this first one, this first matrix here, right. So, this cell actually gives you the value of distance between this G1 replicate, right. So, it is distance with itself, right. So, this will give you like 0 distance, okay. And on the right you can see the color code here, right. So, darker color means lower distance, okay, and lighter color means higher distance, okay. So, 0, so along the diagonal you will see 0 distance because we are comparing that sample with itself, okay. So, there will not be any distance because the same sample. Now, this one here, the next one is comparison between these two ones, right. So, again these are replicates, okay. So, that is why I am saying 4 groups, and you might ask like why do you see 4 G1s, because these are technical replicates, okay. These technical replicates have been done for 4 groups. So, 4 replicates for each group. So, in total 16 samples, okay. And this one, so and so on, right. So, you can see this is the replicate comparison of the replicate. It is actually not very similar, there is some distance, you can see a bit lighter shade. Then you have the comparison of this one with this one, right. So, you have again this one. So, again this is a comparison between this G1 and this G1, okay. For this one, this is distance between these two, right. So, you can now imagine, right, you have these matrix kind of form where you are comparing the distance between each pair, okay. So, you have replicates for each group and then you are comparing distance between each pair. Now, what you can probably notice if you look like at the overall matrix, okay, what you probably will notice is that these G1 replicates actually they are very close to each other, right. So, again this is now clustered. Okay, so which means samples that are close to each other in terms of distance, right, they will be clustered together. Okay, so what you see here is that these 4 G1 replicates, they are clustered together, right. So they are present in this corner of this matrix. Okay, so which means they are actually very similar in expression to each other. And you can probably notice the other cluster, if you look carefully, that there is another cluster here, okay, on this side. You see that, right? So what it means, the rest of the groups are probably very similar to each other. Okay, they are again because they share some transcriptome profile, right. So they have shared transcriptome profile. The genes are very similar in expression across this all sample. So what it means, probably we have 2 clusters, right, one consisting of the G1 samples, right, G1 replicates, and the other one the other groups, right, the other cluster consists of this all these G2, G3 and G4 samples. Interestingly, you have one G4 that actually stands out, right. It is not actually close to G1, it is not also close to the other cluster, because you can see most of these distances are in lighter shade, right, which means there is the distance is high. Okay, so that maybe there was some issue with this experiment of this sample, right, when RNA was isolated or processing of the sample. So this might be an outlier that we might have to discard before we actually go for analysis. All right, so you probably understand now, like it gives us an insight, right. We have 2 clusters, and maybe it is important or it would be interesting to compare G1 versus the rest. Okay, G2 versus G4 differential expression or G2 versus G3 is probably not will not give you some interesting results, because they are probably very similar in terms of expression profile. Okay, so G1 versus rest would be the probably the most interesting differential expression analysis that we can do. Okay, so this kind of insight we can get when we are using this kind of clustering approaches. So coming to the principal component analysis or PCA. So this is again a very not a very detailed discussion. I will just very briefly mention what we do in PCA, but you can find out a lot more. This is there will be a lot more elaborate analysis that you can do with this PCA form. So this is a dimensional reduction technique, but that tries to preserve the information that is present in the data. So when I say dimensionality reduction, so in our data, normalized count data, we have counts for maybe 10,000 genes, right, 7,000, 10,000 or 20,000 genes. Okay, and working with that many dimensions is very difficult. Okay, and maybe some dimensions are not so important, whereas some dimensions are important. Okay, so what principal component analysis tries to do is it will try to identify, right, a combination of these variables that kind of explains the variation that is present in the data. Okay, and it will try to reduce these dimensions, right, from 10,000 to maybe 10 or 100 that can explain the variation that is present in the data. Okay, so it tries to preserve the information as much as possible. Okay, so it derives principal components. So these new variables are called principal components, and these are combinations of original variables, right. So it can be maybe combination of 10 genes or 20 genes, right. So it is looking at, it will identify which gives which can explain the variation better, right. And then of course, once we have this, right, once we identify these principal components, we can see, right, which samples are actually similar to each other and which samples are different. Okay. So we can apply this principal component analysis to the same samples, right. Again, we have this group 1, group 2, group 3, group 4, right, and we have 4 replicates for each group. Okay, and again you can see the colors, right, in red, green, blue, and purple, right. So you can see these 4 colors, and you can probably notice now. So the x axis is the PC1, the principal component 1, and the y axis is the principal component 2. We can have principal component 3 and so on, but usually principal component 1 and principal component 2, they explain the most variance that is present in the data. Okay, so what you probably notice from this plot is this group 1 again stays apart from the rest of the sample, right. This is clear, right. So this is completely separated from the rest of the samples, whereas group 2, group 3 and group 4, they are very close to each other, right. These replicates, these are very close to each other on the other side. Again, one point you probably notice is that there is one group 4 sample that is kind of far away from all these clusters, right. It does not cluster with group 1 or with the group 2, group 3, group 4 cluster, right. So, it is probably an outlier that we may have to remove before we can actually do any differential expression analysis, right. So, because otherwise this might affect the results, okay, and this could be an artifact of experimental errors, etc. Okay, so now once we have done the preliminary analysis, we have identified, okay, which is the most informative comparison, right. What would be the most informative comparison? We see is probably G1 versus G4 or G4 versus G1, right, or maybe G1 versus the rest of the samples. Okay. Now, how do you test for difference in gene expression? Okay, how do you identify these differentially expressed genes? What is the method? Okay. And let us take this example, right, disease versus healthy or condition 1 versus condition 2, that kind of will help us understand the process, right. So, we have let us say gene I, right. So, it could be any value, right. So, again depending on the organism, this I will vary from let us say 1 to 10,000 or 1 to 20,000, right, again depending on the organism, and we do something called hypothesis testing for each gene. Okay. So, this is the general approach, but we will then talk about specific tools, and we will take one tool and we will specifically mention what kind of testing we are doing, what kind of statistical test we use, etc. Okay. So, for hypothesis testing, you might be aware that we need a null model, which we denote usually by H naught, and the null model is that there is no difference in expression between two sets of samples, right. So, if you are talking about disease versus healthy samples, the null model says, okay, there is no difference in expression. Okay. Now, since you have null model, you also have an alternative model which says expression of gene I is different between two sets of samples, right. So, it is an opposite of the null model. Okay. Now, in hypothesis testing, what we will have to do is then we do a statistical test among replicates of this normalized count data that we have generated. Okay. So, in case we want to do let us say G1 versus G4 comparison, we will do a statistical test between the replicates of G1 and G4. Okay. So, this replicate number can vary between 3 to 5. Again, it will be better to have more replicates, but these experiments are expensive, right. Doing the RNA-seq is expensive. So, again we kind of settle for 3 to 5 replicates per sample, right. So, we cannot have more than that usually. So, this is something we need to keep in mind, because this will come to us later on. Okay. So, because the sample number is low, right. So, we have to be a bit careful about the statistical test. Okay. So, when you do the statistical test, I will come what kind of statistical test we can do a bit later, and from the statistical test we get a level of significance or something like a p value that will tell, okay, whether the results are significant, and we have a threshold. This is usually 0.05 and sometimes 0.01. You can be more stringent, you can also set it to be 0.001 or so. Now, the usual threshold is 0.05. So, if p is less than 0.05, then we can reject the null hypothesis. If p is greater than 0.05, then we do not have enough evidence to reject null hypothesis, right. So, this is standard hypothesis testing. Now, when we reject null hypothesis, what does it mean? It means that the gene is differentially expressed. Okay. So, the null hypothesis was that there is no difference in expression of gene I between these two sets of samples. When you reject the null hypothesis, it means that is actually not the case, right. So, we say the gene is differentially expressed between the two samples, right. So, we identify that gene I as a differentially expressed gene, okay, between the two samples. Now, one thing we will keep in mind is that since we are doing this hypothesis testing for all genes, right. So, I could be 1 to 10,000, we need something called multiple hypothesis testing correction. Okay. So, you will see this in some tools, right. This is already implemented. We will talk about multiple hypothesis testing in later classes. Okay. This is something we need to understand a bit better. Okay. So, we do this hypothesis testing for all genes in the sample cells, tissues, right. Again, I can vary from 1 to 5,000, 1 to 10,000, right. So, we do this hypothesis testing for all genes, and then we then identify genes for which we can reject the null hypothesis, right, because these are the genes that are differentially expressed between these two sets of samples. Okay. So, this can be let us say 500 genes or 100 genes, right, and this list will give us the genes that are differentially expressed between let us say disease versus healthy condition. Okay. And as I mentioned, the goal is to understand the biological difference, right, between samples or control versus treatment or disease versus healthy, right. So, these genes can help us understand the biological differences. Of course, we will talk about how we actually better interpret the data once we have identified this set of genes. Okay, so we will talk about that in later classes. There are ways to actually interpret this analysis. Okay, so let us now talk about the statistical test that we do. So we can have something like parametric methods. So I'm not sure if you are aware of parametric methods and non-parametric methods. If not, I'll just mention. So parametric methods, there is an assumption about the underlying data. So here it's the count data, so that we are working with, and there is an underlying assumption about the count data distribution. Okay, so this is normalized count data that we are working with, and there is an assumption, okay, the count data follows certain distribution. Okay, and of course, if your count data distribution differs from this assumption, that might lead to a bit of error in the results. So these results will be sensitive to violations in assumption. So if your distribution is not following that assumption, let us say you assume, okay, your data follows normal distribution, but the distribution is not actually normal, then of course the results will be a bit sensitive and it might give you some false positives. On the other hand, you have non-parametric methods. Okay, so where there is no assumption about the count data distribution, so they are kind of not sensitive to these violations in assumption, etc., because they are not assuming any count data distribution. So they usually are more robust because they can take any form of shape, any form of distribution, and the results will be valid. Okay, so here are some tools that I will just mention that use these parametric methods. Okay, again this is a list, and there are probably even more tools that you can use. Some of them I have just mentioned, for example, the CupDiff I have mentioned earlier. Then you have BASEC, we have DSEC and DSEC2, we have AJAR, Sleuth and EBSEC. Okay, so out of these two, I'll just discuss couple of them in more detail. Then on the non-parametric side, you have NOI-Sec and SAM-Sec. Okay, of course, we will not go into all these tools. We will not discuss all these. I'll just mention some of them. Okay, so what parametric methods in general do is that they assume this distribution for the count data, and you can assume a lot of different types of distribution, Gaussian distribution, so normal distribution you can assume, or binomial distribution, Poisson distribution, or negative binomial distribution. So what researchers have seen is that negative binomial distribution actually best fits the count data because it is usually over dispersed. So the dispersion is quite high. So here mean actually is less than the variance. Okay, so we usually use the negative binomial distribution. So all these tools that I have just mentioned, they use this negative binomial distribution assumption for the count data. So out of these tools, I will discuss DSEC2 in a lot detail. We will talk about the steps, etc. So you will understand how this analysis works, and I will also mention briefly about AJAR. These are the two most popular tools for differential expression analysis. Okay, so for the rest, I will just give you the references so that you can look up and read. So here is the reference for BASEC. Again, the packages are available, the tools are available as packages in Bioconductor in R. So you can download them and install them. So we'll in the hands-on we'll talk about R and how to install packages. We'll install some of them, for example DSEC2, and we'll analyze our data. So similarly for CupDiff, you have the tool available. This is the reference, and you can look up. So again for Sleuth, again the tool is available, the reference is here. You can see and learn more about it. Similarly, you have for EBSEC, again you have the reference and you have the link to the software. For non-parametric methods, you have the NOI-Sec. Again, there is a Bioconductor package for this tool available. You can install in R and you can use this. And finally, this is for the SAM-Sec. This is a tool again employing a non-parametric approach for this differential expression analysis. So let us now go into details of one of the tools. This is a very popular tool for doing differential expression analysis, is called DSEC2. Okay, so the DSEC was the earlier version, the current version is DSEC2. Okay, So, how does this work? So, here is the reference for this tool, and then you have the R package. We will install this tool actually in our hands on, and we will use this tool for differential expression analysis. So, how does it work? Okay. So, this tool will start with count matrix, and where we can see the count will be Kij. Okay. This Ki is the for the gene, right. So, this count is for gene I in sample j. Okay. So, we have, let us say, in our earlier example we have, let us say, 16 samples and we have 7,000 genes. Okay. So, we can see I will run from 1 to 7,000 and j will run from 1 to 16. Okay. And this tool starts with raw count data. Okay. It will not take normalized count data, it takes raw count data, and it considers only uniquely mapped reads. So, if you are giving it raw count data, you should give only the uniquely mapped reads and discard reads that map to multiple genes or multiple transcripts. And for paired end data, the mate should map within the gene. Okay. So, if you are working with paired end data, make sure that both the mates they map within the gene. Okay. If one mate maps outside the gene, you should discard that. Okay. This is something that has been mentioned in the paper, and again, why that is the case, the reasoning is given there. Okay. So, there is a normalization step. So, DSEC2 performs a normalization from the raw count data, and it uses this median of ratios method, which we discussed in the last class, right. And we have seen that this method can correct for difference in sequence depth and to some extent for RNA composition bias. Now, let us move on to the model, right. This is the most important part of the DSEC2. So, as I mentioned, this is a parametric method. So, the read counts are modeled using negative binomial distribution, and what we assume then is that Kij follows negative binomial distribution, or sometimes called gamma Poisson distribution, with mean mu I j and dispersion alpha I. Okay. So, two parameters are there, and then this mu I j is proportional to the cDNA concentration in the library, right. So, which is denoted by Qij, and this is scaled by the normalization factor Sij or Sj, right. So, we have talked about this normalization factor or scaling factor, right. When we talked about median of ratios method, we get a normalization factor for each sample, right. So, here we can use that factor, right, for example, Sj, but sometimes if we want to calculate this normalization factor for each gene separately, right. So, in that case this will be Sij, right. So, this normalization factor is unique for each gene, and then we can supply that as well. Okay. So, just to remind you, I is for gene and j is for sample. So, Sj is a size factor that has been calculated from median of ratios method. This is the default, right. This is what DSEC2 will do. So, just to put it in the notation, so Kij follows negative binomial with these two parameters Sjqij and alpha I. Okay. And using generalized linear model, what it does, it tries to fit this log to Qij with this xjr and beta ir, right. So, summation of this beta are the coefficient values, right, and x are the design matrix element, right. So, for example, it just tells whether this sample belongs to disease sample or belongs to healthy samples, right. So, in our example, it will tell us whether this is group 1 or group 2, group 3 or group 4, and beta is the coefficients. Okay. So, we will take a very simple example that I think would make it even clear. So, in simple experiments, this design part, right, this design matrix, right, it is usually sample status, and you can say this is 0 for healthy and 1 for diseased. Okay. So, something like that, and you can also accommodate more complex designs, right, because this is generalized linear model, you can have much more complex designs, you can have multiple factors, right, not just healthy and disease. You can also might add, let us say you have some treatment, right. For example, in some disease, some patients have some treatment, so you are using that treatment data, and you can also think maybe the age of the patient is also important for this analysis, right. So, you can add age also, and you can see, right, whether which of these factors probably affect the expression. Okay. And you can also have interaction terms, right. So, if you are familiar with linear models, right, you can see like you can use the interaction terms, and this being a linear model can also do that. Okay. So, let us take a very simple design if you are not very comfortable with these complex equations, right. So, we take a simple design and we will understand how this works. Okay. So, we have disease versus healthy design, right, and you can simplify this model as in this way, right, log to Qij, is beta IH plus xj beta ID. Okay. So, we can say xj, this is the design matrix part, right. So, xj is 0 for healthy sample and xj is equals to 1 for disease sample. Okay. And the null model, right, this is where the statistical model comes in. So, null model is beta ID equals to 0, right. So, there is no difference in expression between healthy and disease sample, right. So, this second term here, this becomes 0. Okay. But of course, then you have the alternative model, so which says beta ID not equal to 0, and that means there would be something called log fold change or LFC, that is given by beta ID. So, just note that this is in log, right. So, this is log transform. So, beta ID will give the log fold change, log to fold change, okay, or in short LFC. We will discuss what is this LFC a bit later. Okay, so, what this generalized linear model does, right, it gives us estimates of these coefficients, right, of this beta IR that is present in the model, and that best fits the data. Okay. And so, in that process what you get is the overall expression level for each gene as well as the log to fold change value for each gene between disease versus healthy sample, for example, right. So, you can imagine this, right, the similar concept will apply if you are talking about condition 1, condition 2 analysis, etc. So, now at this model, right, you get this log fold change as well as the mean expression level for each gene. Okay. And there are now the next steps. So, the other step is the dispersion, because the statistical test that you are doing depends on this parameter alpha I, right. So, we need to estimate this dispersion from the data itself, right. We need to do something called LFC shrinkage, and finally, we will also need to do the hypothesis testing. I just mentioned, right, we do some hypothesis testing, right, but what kind of hypothesis testing we have not talked about. Here we have just set up the model, we have estimated the parameters, but we have not identified the differentially expressed genes, okay, which we will do in the next class. So, here are the references for this class. To conclude, so we have seen, right, differential gene expression analysis allows us to identify genes that change expression pattern across different conditions or different samples, right. Here we took examples of disease versus healthy, and we have also talked about some of the preliminary analysis, right, that can help us direct towards more meaningful analysis, right. So, we can choose which groups we should compare when you are going for this differential expression analysis. And we have also talked about parametric and non-parametric based methods for model testing. We have seen that negative binomial distribution best fits the data, and we have started our discussion on differential expression analysis with TSEC2. So, there are some steps remaining which we will be discussing in the next class. Thank you.